Professor Jamieson heads up the eye research group and is arguably the leading researcher in genetics and eyes within Australia. This research is used to highly target treatments and hopefully will improve a wide spectrum of treatments over time. And she's here speaking today about the latest research. So give a hand to Professor Jamieson.
Thank you very much, Matt, and thank you very much for inviting me here today. And I've found it really inspiring as well. And one of the things I've enjoyed all the talks actually, but I really found it great to hear this morning about people finding their passion and seeing how that helped them in their lives and achieving the dreams that they want to do. And it made me reflect a little bit on my journey in coming here and reflecting on the things and how the passion has helped me in the things that I have wanted to achieve. And like Donna, who spoke this morning, I actually came from a very small western southern Queensland sheep farm. I think we're about 9 hours from Brisbane. So a very small school. And I expressed that I was interested in helping people. I was also totally fascinated by what was under our skin, what was behind our eyes, how all those things worked. And I remember expressing that I was interested in doing medicine and interested in helping people as much as I could. And I remember at that time, you're only a kid from a country school, how could you possibly do that? And so I think it was Jodie said, that got burnt into her brain, something like that. And that certainly got burnt into my brain as well. And that really inspired me and to follow my passion, follow my dream, and see how that could lead me in life to achieve the things that I wanted to do and help people in as many possible ways as I could. And so in the research that we're doing at the moment, what we're aiming to do is work with patients with all the clinical features that they have to try and understand what they're showing us in terms of what we can then do to understand where those problems are coming from and how then we can work to understand to give genetic information for families if that's important for them, and then also to work on possible treatment directions to help improve people's health if that's a direction that they'd really like to move in. And so the particular conditions that my research group works on at the moment is particularly genetic eye conditions, which include things like congenital cataract and also retinitis pigmentosa that we've heard a bit about today as well. And so that's the front part of the eye and the back part of the eye. And so in doing that work, we have to think about the particular aspects of the particular disease and how we can work towards what we're doing. And what are the things that might be important for people in relation to this research, which may or may not be helpful to them, but just genetic diagnosis can be a useful thing for people, and particularly I see patients at the Children's Hospital at Westmead and also the Westmead Adult Hospital. And so the various people in those situations have different questions. But I just put up an example here, really. So one of the things that people are concerned about, especially people who might be the only affected person in their family, and they're thinking about having children, they're wondering, so what's the chances of this, happening for my children? They want to be prepared and have that information. And depending on the, if it is a genetic condition, depending on the inheritance pattern, that can be something like less than 1% chance or up to a 50% chance. And so people often would like to know, well, is it actually less than 1% or is it 50%? And if we don't have the genetic information, which in many cases until recently we haven't, and still we don't in a lot of cases, then we can't give people any more information than, oh, it could be less than 1% or it could be 50%. And the other reason is that we would very much like to move towards treatments that might preserve people's vision or prevent further loss, or even give people back some vision where they might not have had some. This is about the genes and chromosomes, which I probably won't go into at this point in time because it is the end of the day. And I do appreciate that you've been here for a rather long time. But you've probably heard, and this is a very busy slide, so I'm not going to go all through it, but you've probably heard in the news about DNA, chromosomes, genes, and that sort of thing. So the main thing to remember is in our cells of our body, if we're thinking about DNA, basically there's 3 gigabytes of data wound up in our chromosomes in all the cells of our bodies, and they code for about 23,000 genes. So that is an enormous amount of data. So for people who've affected with conditions that do have a genetic basis, this is part of the reason why genetic information has been so difficult, because up until now, to look at one particular gene has cost about $2,000. And the problem with these genetic eye conditions, there's often 50 to 100 genes that might cause each particular condition. To get the particular answer, you have to look at 50 genes, $2,000 a gene, $100,000 per patient, 3 months per gene, 12 years for a result. Not particularly helpful. And so there has been a revolution in the genomics and genetics world, and there's been a whole new genomics way of approaching this. And so now you can look at all of those genes at once. So that becomes a test that's more like $2,500 per patient. Still not, of course, Medicare available, still through research studies, still quite difficult, but certainly a lot of progress compared with previously. And of course, I'm not going to go through this, but we're using that technology in genetic eye disorders, and so far we've looked at around about 160 children and adults with genetic eye diseases through the research work. And in congenital cataract, that gives us a detection rate of about, to finding the genetic diagnosis for people who want to know that information, about a 70% detection rate. And then for people with the retinal dystrophy type conditions, about a 60% detection rate. So of course that means that there's certainly plenty of people still out there that the research isn't able to give answers for, but there's certainly quite a lot of progress in this area. I won't go through all that, but basically this is not easy. It takes a lot to work through 3 gigabytes of data and make any sense of it. And then, again, I won't go through this slide either, but basically once we've got that data and we've found the gene, so then the task is to understand how those genes work, what we can do to understand how we can improve the situation so that we can work towards helping with preservation of vision and other potential treatments. And I'll just give you a couple of examples of the type of work that we're doing. So, for instance, this was a patient who had a very severe eye disease. Front part of the eye was quite affected. That person had a what we call a chromosomal translocation, so some of their DNA was swapped around. And so we investigated that and found the new candidate disease gene, which is helpful then, because remember I said that we're not finding the answers for everybody, and that's because there's other disease genes that haven't been found yet. But to understand those disease genes, we've got to prove then that they are a disease gene. So in this particular case, we undertook mouse studies and did a mouse knockout. The mouse got the same condition as the patient had, so that indicates that that is a novel disease gene. So we're going to publish that in the scientific literature. And then the other way that we're working towards understanding genes and working towards treatment is in, this is an example from a retinitis pigmentosa family. So we're using this new DNA sequencing technology to find the novel disease gene in that family, and then we're in the process, in this case, we've again found the novel disease gene, but then we've got to work then towards understanding how that disease gene works. And so this brings into play other new technology which is related to stem cells. So stem cells are particularly of interest, particularly in relation to there's new technologies now where you can actually take skin cells, so take a skin biopsy, culture that, so you get a skin cell culture, and then turn those skin cells actually into retinal cells or even lens cells, different types of eye-related cells. And then that's useful because you can work on that to understand functions about the particular gene or protein that's affected. But if you can actually understand the gene mistake, then you can do something called genome engineering, CRISPR technology, and fix the gene mistake, with the idea being, so particularly for the retinal dystrophies, fix the gene mistake, culture those cells to get repaired retinal cells, transplant that back, and then aim to restore some vision for the person. Now, clearly, that is a lot of work and not an easy road, but I think there is, certainly in mouse models, there is a lot of hope from that sort of technology. And, of course, there are other gene-based approaches, gene therapy you've heard of, and also there's potential here for pharmacological approaches. But again, so pharmacological approaches, that means using a particular drug. Maybe there could be a particular drug that could be, say, for the retinal conditions, a subretinal injection might be particularly helpful. But again, understanding the function of the gene, we need to do that to proceed towards these types of strategies. So there's a lot of there's a lot of work that needs to be done, but there are new technologies, and we've heard a lot about new technologies just recently, but there's new technologies that are also available in the research world, and I think that can give us quite a bit of hope that these sorts of things will eventually be helpful for people. And so I just want to end by letting you know that, of course, there's resources at Save Sight Institute, as of course we know from today. And then if there's particular genetic services that you're potentially interested in, there also throughout New South Wales, and you can access those by going onto that website, which is genetics.edu.au, which can let you know the particular genetic service that's nearby where you live. So thank you very much, and I hope you've enjoyed the day as much as I have, because I've found it really fantastic and inspiring, and it's been great to meet many people as well. Thank you very much.
Thank you very much. Thank you, Professor.